Welcome to This Explainer. Today, we're literally putting on our detective hats to investigate one of the absolute greatest whodunits in the history of biology. For a long time, humanity knew that traits were passed down from parents to offspring, but the actual physical molecule responsible for this, the physical blueprint of life, was a complete mystery. So we're going to examine the exact case files and historical experiments that finally unmask the molecule of inheritance.
Okay, let's dive into this case file. We'll be investigating in 5 phases: the mystery of inheritance, Griffith's transforming principle, purifying the genetic culprit, the blender experiment, and finally, unlocking the structure.
Part 1. The Mystery of Inheritance.
Before we could unmask the genetic material, scientists actually had to build a profile of their suspect. Early biologists knew this mystery molecule had to meet 4 incredibly strict criteria. First up, storage of information. It had to hold the massive, complex instruction manual for an entire organism. Second, replication. It needed some mechanism to be copied flawlessly so it could be passed on to the next generation. Third, expression. It had to actually do something, right? It had to control cell structure and function to produce physical traits, or phenotypes. And fourth, variation. It had to be capable of mutating. This is absolutely crucial because it allows for the natural variation that drives evolution. Our timeline begins with a few early clues. Back in 1866, Gregor Mendel figured out that traits are inherited in discrete units, which we later started calling genes. Just a few years after that, in 1869, Friedrich Miescher isolated this strange new substance from, get this, pus cells on discarded surgical bandages. Yeah, gross, but a major breakthrough. He called it nuclein, which we now know as DNA. But here's the thing, at the time its function was a total mystery. In fact, by the early 1900s, the entire scientific community heavily favored proteins as the prime genetic suspect. Proteins are incredibly chemically diverse, while DNA just seemed way too simple to hold all that complex information. So how did we finally exonerate proteins and look closer at DNA?
Part 2. Griffith's transforming principle.
Let's jump to 1928. Our first detective, Frederick Griffith, was working with Streptococcus pneumonia bacteria, and there are 2 really distinct strains to keep in mind here: the S strain, which is virulent and deadly, and the R strain, which is non-virulent and completely harmless to the mice.
So Griffith ran a series of tests, and the first few went exactly as you'd expect. The live S strain killed the mouse. The live R strain left the mouse totally unharmed. And heat killing the deadly S strain made it harmless. But the critical, mind-bending moment happens in step 4. Griffith mixed that harmless dead S strain with the harmless live R strain. The result? The mouse died! No way, right? Even more shocking, when he examined the dead mouse, he found live, deadly S strain bacteria inside it. He concluded that some sort of transforming principle had literally jumped from the dead S bacteria and physically changed or transformed the living R bacteria into killers. But what on earth was this principle?
Part 3. Purifying the genetic culprit. To answer what exactly that transforming principle was, we need to fast forward to 1944. Avery, McLeod, and McCarty took over the case. They took Griffith's deadly S strain and extracted its chemical components into 5 separate test tubes. Going right down the line, they used specific enzymes to destroy just one chemical component per tube, destroying the polysaccharides, then the lipids, the RNA, the proteins, and finally, the DNA. They then mixed these purified extracts with the harmless R strain and injected them into mice. They were basically systematically waiting to see which mixture would still cause that deadly transformation.
So results here were a massive breakthrough. When they used an enzyme to destroy the RNA or the proteins, the mice still died. The transformation still happened. But when they used DNase to destroy the DNA, the mouse lived. Okay, so the absolute most crucial point to take away here is this. Only when the DNA was destroyed did the transforming ability vanish. This was our very first piece of concrete proof that DNA, not protein, was the transforming principle, the actual genetic material.
Part 4. The blender experiment.
Now, despite Avery's brilliant work, skeptics still kind of clung to the protein theory. We needed an irrefutable smoking gun. So in 1952, Hershey and Chase devised this incredibly clever methodology using a virus that infects bacteria. Viruses are essentially just DNA wrapped up in a protein coat. They created 2 batches. In the first batch, they used radioactive phosphorus-32 to label the virus's DNA. In the second batch, they used radioactive sulfur-35 to label the protein coat. They let these phages infect the bacteria, put them in a literal, ordinary kitchen blender to aggressively knock the empty virus shells off, and used a centrifuge to separate the heavy bacteria at the bottom from the lighter viral shells, the supernatants, at the top. The results of that centrifuge spin officially solved the case. The radioactive phosphorus, the stuff tagging the DNA, was found inside the bacterial cells, right at the bottom of the tube. The radioactive sulfur, tagging the protein, stayed completely outside in the surrounding liquid. And this just brilliantly illustrates the final proof. Viruses actually inject their DNA into the host to direct virus replication, while the protein coat just gets left behind like an empty wrapper. Protein was fully, definitively exonerated. DNA was our actual culprit. So, just synthesizing our case file sequentially here, from memory. In 1928, Griffith showed us that traits can be physically transferred. In 1944, Avery's team proved chemically that DNA causes this transformation. And in 1952, Hershey and Chase proved DNA actually enters a cell to dictate cellular machinery. The chain of evidence is honestly flawless.
Part 5. Unlocking the structure.
Okay, so now that the scientific community finally agreed DNA was our genetic culprit, a new problem emerged. How did this molecule meet all those complex criteria we talked about at the start? We needed its physical profile. The first big structural clues came from Erwin Chargaff in 1950. He discovered two absolutely vital things. First, he found that in any DNA sample, the amount of adenine always equals thymine, and guanine always equals cytosine, so A equals T and G equals C. Second, he discovered that the overall composition of these bases varied wildly between different species. This was huge. It meant DNA wasn't just some repetitive, boring chain. Its variation held massive informational potential.
But what did it actually look like in 3D space? In 1952, Rosalind Franklin pointed an X-ray beam at crystallized DNA and captured the iconic Photo 51. To a trained crystallographer, that striking X pattern radiating from the center is undeniable, physical proof of one very specific shape. DNA is a helix.
Using Chargaff's rules and Franklin's unbelievable X-ray data, James Watson and Francis Crick built their famous 1953 model of the DNA double helix. It was just a beautiful moment of scientific synthesis. The structure they proposed, two twisting strands held together by complementary AT and GC base pairs, perfectly explained everything. The sequence of the bases explained how information is stored, and the paired strands explained exactly how DNA could literally be unzipped and flawlessly replicated. The profile perfectly matched our suspect.
We've officially closed the case on finding the molecule of inheritance. But as we look at this blueprint of life, I want to leave you with this thought. Consider the sheer mind-bending scale of it all. How does this elegantly simple helical structure, using only a four-letter chemical alphabet, manage to hold the vast, infinite informational potential for all living variation on planet Earth? From a microscopic bacterium to a towering redwood tree, all the way to you and me, it all comes down to the beautiful geometry of DNA. Keep questioning, keep learning, and thank you so much for joining me on this explainer.